I'm Xina.
I'm Rohit.
And I'm Harsha.
And we're proposing using a CRISPR-based workflow to study the soil microbiome in order to evaluate plant health.
Our astronauts continuously state that what they missed the most in their time in space was eating a fresh salad. Eating freeze-dried food every day, our astronauts miss out on key nutrients that we normally consume on Earth. But the ability to grow fresh plants in space solves so many more issues. Plants can provide psychological relief to astronauts, naturally purify the air, and it's the first step we can take before colonizing any future planet. In light of all these reasons, scientists have studied how plants can be cultivated in space since the dawn of space travel 80 years ago. But in 2021, exciting news of the discovery of 4 strains of a plant growth-promoting bacteria being on board the ISS shows great promise that it can be harnessed as a tool to help plants thrive in space.
Plant growth-promoting microbes interact with the roots of plants and also exist readily in the surrounding soil. They're the plant's unseen underground support system. Without these microbes, plants would have a harder time functioning optimally. These microbes express genes that produce proteins that help the plant fixate nitrogen, enhance root functioning, and also aid in phytopathogen resistance. As a result, we are focusing on 2 of the genes that we think would be most beneficial to plants under the adverse conditions found in space. These 2 genes are ACDS and IIH. ACDS produces an enzyme, ACC deaminase, which helps to decrease plant stress and also helps to overcome drought or radiation-like conditions. Similarly, IIH helps to improve root health and also helps induce pathogen resistance by producing a protein that aids in the synthesis of auxin indole 3 acetic acid.
However, it is difficult to study gene expression in space because samples must be brought back to Earth for analysis, which can take up to 6 months. Extensive sample preparation and the need for trained personnel to conduct RT-qPCR prohibits faster progress in isolating and studying these beneficial microbes on the ISS. Therefore, an integrated microbial monitoring system that can process and analyze samples in flight is necessary. Our study will be able to develop a biosensor to achieve safe and sustainable space exploration. Our proposed workflow can quantify gene expression in plant growth promoting rhizobacteria with the same sensitivity and specificity as RT-qPCR, but will not need RNA extraction, can take extremely crude samples, and can be completed as a 1-tube reaction with fluorescent readout in under an hour. Our workflow will be utilizing CRISPR-Cas13 to study gene expression of the soil microbiome to understand plant health. CRISPR-Cas13 can bind to RNA transcripts of interest through a well-established mechanism used in viral diagnostics here on Earth. And so as a result, it's already developed to be fast and convenient. We can do this with just the Genes in Space toolkit with little to no training required. Our workflow, by utilizing CRISPR-Cas13, we hypothesize that this workflow will see IAH and ACDS downregulation in space due to the increased plant stress observed in spaceflight conditions. Now, let's look at the components of our workflow.
RPA is a much simpler alternative to RT-PCR. In RPA, we will take the RNA from our soil sample and we will convert it to DNA through a reverse transcriptase. This DNA, which is much more stable, can then be amplified through the RPA process. Then we will convert the DNA to RNA again through a polymerase, which we can run through our Cas13 sensing. This entire process can be completed at 1 temperature and in less than 30 minutes, including our Cas13 sensing assay.
After our RNA is amplified, our guide RNA Cas13 complex will then bind to our target RNA. Then, our Cas13 enzyme will be activated to non-specifically cleave our fluorophore-quencher probes, which will then provide us with a quantitative fluorescent readout of the gene expression in our soil samples. Now, for our ambitious experiment, we're going to split our workflow into 2 objectives. First, we want to determine how well BioBits can synthesize a working CRISPR-Cas13 enzyme in space. And second, we want to determine if IAH and ACDS will be downregulated in a space microbiome.
To evaluate the efficacy of cell-free synthesis of CRISPR in space, we will design a plasmid vector. This plasmid vector will encode for a guide RNA Cas13 complex that I will refer to as Cas13RNP, and this will be our biosensor. It will also encode for red fluorescing protein, or RFP, to visualize successful plasmid expression. And so the level of red fluorescence will serve as a measure of Cas13RNP expression and yield. We will set up 5 BioBits reactions, 3 controls, 1 for green fluorescence, 1 for red fluorescence, and a negative control of just water, and 2 tubes in sequence that will both contain our Cas13RNP plasmid and fluorophore quencher reporter. And for the second tube, we will also add an RNA strand that is actually complementary to our guide RNA. And so, as you recall, once it can find this complementary RNA strand and it binds to it, our Cas13 RNP will become activated to cleave those fluorophore-quencher reporters, and it will result in a green fluorescence. Afterwards, we will set up a standard curve of known RFP concentrations, and so for the first tube, we can quantify our Cas13 RNP yield by comparing it to our RFP standard curve. And for our second tube, we will actually assess the functionality of our BioBit synthesized Cas13 RNP. And so then on both Earth and in space, we will set up our tubes. We will then track the fluorescence of our kinetics of our reaction every 30 minutes for 8 hours, normalize and quantify the images, and compare our results. We expect to see similar results on Earth and in space, where for our first tube, it will continue fluorescing red in the presence of, and as it goes towards the end of the assay. And for our second tube with the complementary RNA, because it will actually activate our Cas13 RNP to cleave those reporters, over time we will observe a transition from red to more green fluorescence.
Once we've established that our Cas13 RNP is functional in space, we can use it to detect the gene expression levels in space as well. These 5 tubes here will serve as a calibration curve for our assay, correlating the amount of green fluorescence to the amount of gene that is present in our sample. We will also have a positive control with our activated RNA probe and a negative control with just water, like in our previous trials. We will have 2 identical soil samples that we collect, but we will only include the Cas13 RNP in one of these soil samples. In the second soil sample, we will not include the Cas13 RNP in order to monitor for any background fluorescence that could impact the quantification of our assay. Then, in both Earth and the ISS in parallel, we will track the fluorescence of our assay every 5 minutes in order to ensure that our Cas13 is just as efficient in space as it is on Earth. At the end of an hour, we will monitor the ending fluorescence in order to see how the microbe gene expression profile varies between space and on Earth.
We hypothesized that both IAH and ACDS will be downregulated in space. IAH because plants in space tend to have a weaker immune system, and ACDS because plants in space tend to have higher levels of ethylene, a plant hormone that indicates increased plant stress. And so we expect that the vials on the ISS will fluoresce less green than the vials on Earth, indicating that both of these genes will be underexpressed. Now, for our freeze and fly experiment, we're going to condense both our ambitious objectives into a set of 16 tubes, where half our experiments will take place on Earth and the other half in space. On Earth, we will prepare our RNA soil samples and load it into our Cas13 BioBit system, and immediately freeze it to be sent to space. In space, astronauts can simply thaw our vials and visualize the fluorescence over time with the P51 fluorescence viewer. And just like our ambitious experiment, we will have 2 vials to assess the functionality of our Cas13 RNP system, and 5 vials that will serve as a calibration curve to normalize and quantify gene expression levels in our sample.
In essence, we're proposing a Cas13 RNP system that can monitor gene expression in space without any Earth intervention or complex instrumentation. We're going to use BioBits to synthesize the Cas13 RNP and the fluorescence viewer to heat our samples, as well as monitor our fluorescence output real time. This process only requires pipetting and provides a quick readout. This is the first time that RT-RPA and Cas13 will be sent to space, and because it has been well studied on Earth, we're confident that it will work in space as well. This will help pave the path toward more complex gene studies in space in the future.
In May of 2022, scientists discovered that plants are actually able to grow in extraterrestrial soils, including moon regolith. By studying plant growth-promoting bacteria gene expression in space, we can better optimize these soils to sustain plant growth and equip us on a journey to Mars. Moreover, our biosensor can serve as an important tool to study rural environments on Earth without access to expensive instrumentation.
In a broader scope, we hope that more Cas13 RNPs can be created that target other genes of interest so that we can obtain a full gene expression profile of the plant microbiome. And just by changing our guide RNA sequence, it is easily adaptable for many other uses, such as identifying other microbes of interest for rapid disease diagnostics, and also our experiment sets a precedent for cell-free CRISPR synthesis in space that can enable on-demand gene editing on the ISS. Our workflow will be one of the first steps in replacing expensive instrumentation or trained biologists, making studying living systems possible anywhere and anytime.
We would like to thank our mentor, Kaylee, and the Genes in Space team for giving us this amazing opportunity, and we're open to questions. Thank you.